Hi everyone, we're looking at chapter 15, which is all about how genes make these things called proteins. This is sometimes called the central dogma of biology. Dogma means, a really important part of something or the main idea of something, and so this is a very important fundamental process happening in biology. It looks like this. There are kind of two important processes involved in it. And to illustrate what those processes are, I've kind of drawn or included a figure of a cell here. Keep in mind this is a eukaryotic cell. This process, the central dogma, happens in eukaryotic cells and prokaryotic cells. It happens in all cells. So don't be misled by the fact of how I've included a figure of just a eukaryotic cell. It happens in both. Okay, but in either case, how it works is it starts off with DNA. And DNA is made into this molecule called RNA. Now there are three types of RNA. Regardless though, this is a process known as transcription. The three different types of RNA are the following. There's mRNA, there's rRNA, and there's tRNA. Now I've included these in parentheses down below, not because they're not as important, but because of their purpose. mRNA, what its job is it stores information that is on DNA. And it uses that, it brings that information temporarily, it stores it, and it brings it to a different part of the cell where it can be made into protein. rRNA and tRNA, their job is less to store information. Their purpose is that they are more structural molecules that have a specific function. So rRNA, it is made and it actually folds upon itself and gets converted into ribosomes. So a majority of the structural material from which ribosomes are built is this long ribbon of nucleic acid called rRNA. rRNA is made specifically right here in the nucleolus of the cell, and because that's the place where ribosomes are made. tRNA is going to be a molecule that has an important feature to it because it's a molecule that can bind two things. It can bind mRNA, but it can also bind amino acids. And so because of that special property, this is a molecule that helps us bridge the link between nucleic acids and amino acids, which that is to say it bridges the link between the information stored in the genome and the actual building blocks that make proteins, which are amino acids. Okay, you'll understand what these do, or specifically what this does a little bit later. But in either case, the production of mRNA always is going to be made from DNA. Now you might ask, why is there two steps? Why can't DNA just do these things? Why can't DNA form ribosomes and DNA be a molecule that binds to amino acids? And why do we, if information is already stored on DNA, why do we have to copy it again onto this molecule called mRNA? That seems kind of like a redundant process. Well, the answer is that DNA, specifically the genome, is very large. There's 3 billion about base pairs. So that means there's about 6 billion letters altogether, or nitrogenous bases would be the actual thing that they are, but we sometimes call them letters because it's easier to understand. Okay, and that is all found here in the nucleus of a eukaryotic cell. In a prokaryotic cell, it's kind of sprawling around in the cell. It's not localized in one specific nucleus. But in this example here, imagine that there is 46 chromosomes all unwound and kind of sprawling around in this space. And it's just really, 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 really long molecules of nitrogenous bases in a row. So A's, T's, C's, and G's is how we would abbreviate them. Well, inside this nucleus, all of the information is required to build, to build you, which is pretty fascinating, right? When you think about all of your organs and how complicated they are, and all of the blood vessels passing through your body, and all of the bones that keep your body upright, and all of the muscles that attach to those bones, and all of the neurons and nerves, and all of the small tubing that's in your urinary system and your reproductive system and your digestive system. That's just lots and lots of organs, lots and lots of stuff. All of that information required to build that is found right here in the tiny, teeniest little space of a tiny, teeny little cell. So it's really, really tightly packed information. Now, the information is stored here, but the information is not the final, endpoint. The information is only a means by which protein is made, and it's actually the protein that is made which helps give characteristics to our cells, and if we can, lend different characteristics to different cells, we can produce different tissues, and from those tissues, different organ systems can be made. So it all kind of stems from here. Now, consider, for example, consider, for example, that this might be let's say it's a cell in the pancreas, all right? One of the things that pancreatic cells make, specifically pancreatic cells that are found in the pancreatic islets, is they make a hormone called insulin. Well, insulin is a protein, and it is made from the information that's stored in here. So keep in mind there are 3 billion base pairs in here, and the gene that codes for something like insulin is probably, I don't know what it is off the top of my head, but it's probably on the order of hundreds or thousands of base pairs, not hundreds of thousands, millions, or certainly not billions of base pairs. So if a cell such as this, which we're pretending is a pancreatic islet cell, wanted to make insulin, it would have to go in and actually find the gene, meaning the small region of DNA that holds the information to code for the insulin protein. It would have to go in there, and it would have to actually make kind of like a photocopy of it, so that that photocopy could then be sent out to the different parts of the cell that are responsible for building the protein insulin. So if you imagine that the nucleus is kind of a library, of 100 or 200 really, really thick books, all full of letters, you can imagine that the mRNA and also the tRNA and rRNA are like small photocopies of one page of one of the 100 or 200 books of information. Okay, and so it makes it a lot easier for the cell to build stuff from this really, really, really daunting, packed storage locker full of information. So the cell might go in, it might locate, the 100 or 1,000 base pairs for the insulin gene. It might make a small photocopy of it, and then that small photocopy is small enough where it could go through these little nuclear pores found in the nucleus, and then it could go out to these little red dots, which are ribosomes. Well, ribosomes are the places where protein is built, and so in order for the protein to actually be made, the information or the photocopy of the DNA has to get out to the ribosome. Okay, so these small little pieces, which we would call transcripts, are a lot more mobile. They can get into and out of the cells, nooks and crannies, a lot faster, and certainly are able to make their way to places that the genome would never be able to make it to. So think about transcripts as being photocopies of DNA. Right? The thing that makes them is called transcription. And then once that photocopy makes its way to a ribosome, then the actual process of building the thing that the information is coding for happens. So the instruction manual, so to speak, has to make its way to the site of protein manufacturing, and once it does, then this process occurs, which is known as translation. So these are your big important terms to know here: transcription and translation. Translation is the process of making protein from the photocopy. Transcription is the process of making mRNA from the whole genome library of DNA. Now we'll take a look at transcription in more detail first. Transcription looks a little different in prokaryotic and cells and eukaryotic cells. There are quite a few similarities, but there's also a few differences, which I'll highlight in a little bit. But we'll start off with a eukaryotic cell just because we've just come off of this beautiful, nice visual here, which is of a eukaryotic cell. To understand what the process of transcription looks like, I always find it's helpful to kind of imagine a chromosome. Now, keep in mind, chromosomes look a little bit different in eukaryotic and prokaryotic cells. In a eukaryotic cell such as ours, we have 46 chromosomes. And also what they haven't drawn here, which is a shame, is the histone proteins. So keep in mind that DNA is tightly packed together into these things called chromosomes to help achieve that compaction. There are these histone proteins, which the DNA strand, this is a strand of DNA right here, this strand of DNA will wrap around the protein molecules, wrap around to form these things called nucleosomes. Those all get super tightly packed together. Okay, so the histone proteins are not shown here. Let's keep that in mind. All right. Well, somewhere along this particular chromosome, there's one of the chromosomes in our genome bears the insulin gene, and somewhere in that chromosome is the specific little spot on the chromosome that the gene is found. And when a cell like a pancreatic islet cell needs to make insulin, it has to go out and to figure out which chromosome bears the gene and where specifically on the chromosome the gene is found, and then once it's located, it can copy it. Now, in order to aid its location of the gene, there are these things called promoters. So the first step in this process is initiation. This is finding the gene and then recruiting the proper cellular machinery required to copy it. The place that it's found is going to be, or there's a place that usually is a spot that's just upstream from the gene called the promoter. And the promoter is the spot where all of the cellular machinery is going to be recruited to when the gene needs to be made. And most promoters in all cells have a common feature, which is known as a TATA box. And a TATA box can vary, but all of them are going to have this sequence TATA. So somewhere along this strand of DNA, there will be this little sequence right here, TATA. Now it's significant because these bonds right here, A's to T's or A's to T's, they are a little bit more unstable when changes in temperature and things like that happen, so they can be broken apart just a little bit easier. And so in order for all of the machinery to get into a strand of DNA and copy it, first the strand has to unwind and it has to loose, the two strands have to separate. Remember, DNA that is double-stranded. Here's a strand and here's a strand, and here they're pretty closely associated with one another. But in order to copy the gene, we have to open up the strand. Well, a good place for that is the TATA box because of these loose associations between these base pairs here. And then usually what happens is that there's a TATA box and then a couple of other sequences, and that's going to recruit a bunch of what are called transcription factors. Here's a picture that the book has kind of showing this. Here they're showing a common eukaryotic TATA box, which looks like this, TATA with another couple A's. They're showing this as 30 base pairs upstream from the actual gene of interest that needs to be transcribed. And then in different parts upstream from that, there can be lots of different combinations of promoters. Again, one of the common things is they all have this little TATA box in them, but they all are going to have different types of things specific to the gene. Well, what's important is that the TATA box, it recruits things called transcription factors. And what transcription factors are, they're just basically proteins that either encourage transcription. They would do this by recruiting the cellular machinery required to make RNA. They could, or also, as opposed to encouraging transcription, they could silence transcription. So these transcription factors, they either help the cell make something or not make something, and whether a cell needs to make something or not make something is very important. If your cells just started making insulin, you'd probably be dead in a matter of hours if they couldn't shut that process off. And so your cell needs to, at one point, make insulin and then stop making insulin. And if it made too much of it or too little of it, you would not survive. And so that's probably the case for almost every protein in your body. It needs to be made at one point, and then it needs to shut off. It can't continually be made. Well, the transcription factors are one of the ways that regulate when things are made or not. The way that they work is that they will bind to the promoter regions on the DNA sequence, and there can be a whole bunch of them that bind. And then their main job is they recruit the cellular machinery that's going to do the transcription. The cellular machinery is called the RNA polymerase. There's a couple of different kinds of RNA polymerase. We'll talk about that in a minute. But RNA polymerase is this big protein which is going to be the actual thing that adds RNA molecules together. The name of this molecule literally means the molecule that, or the enzyme that builds a polymer of RNA. Okay. So that's the Tata box, that's the transcription factors, and that's the purpose is to recruit the polymerase. Okay, so getting back here, we see the Tata box. So if this was in fact a pancreatic cell that was needing to make insulin, it would have recruited a bunch of transcription factors to bind to this one little spot on the DNA. Once all of those bound, the RNA polymerase would then come and find those transcription factors and attach to them. That's one of the biggest hurdles in this whole process, is recruiting the RNA polymerase, the actual machine that's going to build the RNA, to the appropriate site at the appropriate time. That's done with the use of promoters and transcription factors, in part. Okay, so once the RNA polymerase is there, then we can begin what's called elongation. Elongation basically just means that the RNA polymerase is going to travel along what we would call the template strand of DNA. So the template strand of DNA would be this one here. And what it does, it's really fascinating. I still can't even comprehend how a simple, small little protein can figure out how to do this. But as the RNA polymerase reads the template DNA, it will add RNA nucleotides to produce a complementary strand, or a strand that is complementary to the DNA template. So you can see, like here, after it bound to the Tata box, it probably read this C, so it added a G. And then it read this G, so it added a C. And then it continued and it read this G, so it added another C. And then it read this A, and then it added a U. That's one of the big differences between DNA and RNA. Recall, RNA doesn't have T's in it. The T's instead are replaced with uracil, so you get U's. Okay, so keep that in mind. Okay, then it read another T, so it added an A. It read another T, so it added an A. It read another A, so it added a U. And it just goes on and on and on and on until, lastly, I guess we're not quite there yet. It goes on and on and on until it finishes transcription, a process known as termination, which I'll talk about in a second. In eukaryotic cells, the type of RNA that is made is determined by which RNA polymerase is used. And specifically, there are 3 different kinds of polymerase enzymes in eukaryotic cells. In prokaryotic cells, there's just one. And they will make these 3 different kinds of RNA. So if it's an mRNA polymerase 1, it makes rRNA. If it's an RNA polymerase 2, it produces mRNA. So this one right here, in this scenario, we're considering this to be a mRNA transcript of a insulin gene. So this would be RNA polymerase 2. And then if it was an RNA polymerase 3, it would make tRNA. So that's where we get our 3 different kinds of RNA from. And then lastly, termination. So the RNA polymerase is going to keep going along the DNA template until there's some sort of signal that tells it to terminate. Now, it's a little bit different. In a eukaryotic cell, sometimes the RNA polymerase will actually, let's say that the information required to build the insulin gene stops here. It would copy all the way up to the gene, and then it would actually keep going and copy a whole bunch of nucleotides beyond the gene, even up to 1,000 or 2,000 base pairs beyond the specific gene, and then that last part would be cut off and modified later. I don't know why it's done this way, but that's just kind of how it works. So it just goes past the endpoint and just keeps going until eventually the RNA polymerase falls off. In prokaryotic cells, it's a little bit different. There's a couple different ways that actually will signal a specific point where the RNA polymerase should fall off. But in either case, whether it's eukaryotic or prokaryotic, the termination involves the RNA polymerase disassociating or falling off of the DNA template strand. And then what we're left with is an RNA transcript that was made from the DNA template, and then the RNA transcript is free to kind of move to a different part of the cell wherever it needs to go. Now, one thing that's interesting is that the transcript that's made from the template is what we would call pre-mRNA if it was, if we're talking about mRNA. This would be different if it was rRNA or tRNA. But for mRNA, we would call the transcript that's made right after transcription, we would call that the pre-mRNA. We call it pre-mRNA because it needs to be modified before it can actually be used to make protein. We call this mRNA processing. This is really only found in eukaryotic cells. And to give an example, consider this little figure here, which is illustrating a pre-mRNA strand. Okay, so imagine this is a ribbon of nucleic acid. Well, this figure is trying to show you that there are different spots of different types of genetic information along a pre-mRNA. There are things called exons, and there are things called introns. And an exon is a part of the mRNA transcript that is expressed. This is the way that I like to think of it. And I remember that E for expressed stands for E, exon. And what expressed means is it means that it will actually code for production of protein. There are other parts of the mRNA called introns that do not code for protein. And these are kind of an interesting thing because they used to be considered junk RNA. So what ends up happening is the cell will actually remove these from the mRNA transcript. And so it was kind of thought that they don't really do anything. But and this is called intron splicing or RNA splicing. But what's kind of interesting is it was found that the cell can actually sometimes remove one set of the introns, another time it can remove another set of introns, and so it can change how many introns are removed in the order that they're removed. This is called alternative splicing, and it yields different mRNAs, mature mRNAs. And so one DNA template can make one pre-mRNA, but that one pre-mRNA can actually be kind of spliced into lots of different mature mRNAs. But in either case, the introns are generally removed and they generally don't code for protein. Also on the pre-mRNA, there will be what are called untranslatable regions on either side. These are spots that do not code for protein as well. And then once this is all spliced out, it looks something like this, where you have a solid region of exon linked together. So this will all code for protein because it's all made from the exons, all linked, together like this. Okay, all of those will get linked together. And then the untranslatable regions are kept intact. They kind of mark the beginning and the end of the actual thing that should be coded for. And then there are a couple other things added. There's what's called a 5' cap and a poly A tail. The 5, as well as the poly A tail, they help to stabilize the molecule of mRNA. They stabilize the transcript. We would call this the mature mRNA because it's been processed. Okay, so once this thing has been processed, it puts these little regions on there to help protect it, because the mRNA, the mature mRNA, is going to have to make a journey out of the nucleus and somewhere into a different part of the cell. It's going to encounter lots of enzymes and organelles and chemicals and molecules as it makes its journey through the cytoplasm, and so these little regions help to stabilize it as it makes that journey. The poly A tail also helps the mRNA make its way out of the nucleus, helps it find its way to a nuclear pore and export out of the nucleus. And the 5' cap is going to help it attach to a ribosome. Another thing that is done is that the RNA is coated with RNA stabilizing proteins. They're going to protect the RNA from damage and being exported to the nucleus. And then the 5' and 3' ends are covered again with the 5' cap. It's going to protect it, help attach to the ribosome, and then the poly A tail protects it, and it signals the export of stuff to the cytoplasm. So it signals the export of this molecule out of the nucleus and into the cell. Okay, those are kind of the summary of what I just mentioned, all the different things involved in mRNA processing. Once that mRNA is processed into a mature, what we'd call now a mature messenger RNA, it can then be used to make protein through the process of translation. Before we talk about that, though, I'll quick mention some of the differences of transcription when it comes to prokaryotes. Some of the big differences here with transcription in prokaryotes is that rather than having 3 different polymerase enzymes, there's just a single polymerase, and it's actually a little bit smaller. It's not made out of as many pieces, only 5 units, subunits instead of 10 subunits like the eukaryotic polymerase enzymes. It also has a specific termination. Remember when we were talking about eukaryotic transcription, the termination phase, it can actually, the polymerase can go way past the end of the gene of interest, and then the later parts are removed. Well, here there's actually a specific termination spot, and the termination can be either called row dependent or row independent. When it's row dependent, it involves this protein called the row protein, and how it works is that this was a DNA template, and there was some polymerase enzyme which went and attached to the DNA template and then started reading the template and making mRNA, or making RNA. Well, there's this little row protein which actually will attach, and it will follow the polymerase as it continues going along the DNA template. There will come a spot in the DNA template where there's a whole bunch of guanine bases, G, 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 and that makes the polymerase kind of get caught up here. It will start to stall and get a little bit sluggish because it takes more time to add cytosines to the guanines, and that will give enough time for the row protein to actually catch up and collide with the RNA polymerase, and then when it collides, it will actually bump it off. So that's what we mean by row dependent. A row independent is a little bit different. There's a place in the DNA template which is a whole bunch of G's, C's, G's, G's, C's, G's, and C's alternating, and that will actually cause what's known as a hairpin in the transcript, meaning it will kind of, the big DNA strand will kind of kink like this, and that will be a thing that knocks the polymerase off. So RNA independent involves the formation of what you could say is a hairpin structure. It's a bend in the nucleic acid, and then the RNA dependent requires the row protein. So this enables transcription to terminate at a pretty specific spot, and the polymerase doesn't keep going on and on and on and on past the gene. That's another difference. What's also interesting is that translation occurs before the termination of transcription. So previously I said that transcription happens and then after that translation happens, but in prokaryotes the two can kind of happen at the same time. Mostly because of the fact that the DNA is kind of found throughout the entire cell. It's not localized in a specific compartment as it is in a eukaryotic cell. In a eukaryotic cell, all of the genome is in the nucleus. In a prokaryotic cell, all of the genome is in a single chromosome that's kind of sprawling throughout the entire cell. So there's a lot more room and there's a lot more access for ribosomes to get in there. And so this can look something like this when it's done. There can be different places where mRNA is made, as you know, let's say there'd be a polymerase reading a template strand and producing with it an mRNA strand. And then right as the mRNA strand is being transcribed from the DNA template, little ribosomes are recruited and they start passing through the mRNA transcript and making protein through the process of translation. We'll talk about this process of translation in greater detail in just the next slide or so here, but for the time being, this is interesting because if it was a eukaryotic cell, the mRNA would actually break off and then travel elsewhere, but here translation is happening right at the site of transcription. And then lastly, in prokaryotic cells, there is no mRNA processing because the mRNA doesn't have to get out of the nucleus and travel very far to get to a ribosome. The ribosomes, in effect, kind of are coming right to the site of transcription to be right there to make kind of an instant protein. All right, next comes translation. So this, again, recall, was the way that mRNA is going to be used to make protein. This is going to happen at the ribosomes because the ribosomes are the site of protein synthesis. Usually it's a chain of amino acids that are 50 to over 1,000 amino acids long. That's what a protein is. And this is going to be a process that consumes more energy than any other process in the cell. Most of what is in the cell is proteins, and so the production of proteins takes up a lot of energy. Proteins pretty much are going to perform every function in the cell. There's proteins that are not only structural, that make up the cytoskeleton and the structure for the organelles and things, but then there's lots of enzymatic proteins, and then those are the proteins that then in turn make all the other things that the cell needs, like phospholipids or cholesterol or glucose or glycogen. All that other stuff is made in essence through enzymes. So proteins pretty much are going to be indirectly or directly related to every single thing the cell does. Now up to this point we've talked about DNA and proteins, but we haven't really specifically talked about how the two are related, how we get from a bunch of letters like A, C's, T's, and G's to actual structural building materials like amino acids assembling together to make a structural or enzymatic type molecule. So the link between those is what's called the genetic code, and maybe you've heard of this term before. What it tells us is it tells us what the letters mean, what they code for. So recall on our transcript here we have ending up with a bunch of letters. The reason why we have all of these letters is because they were copied from the letters that were stored on DNA. Well, those letters mean something. They code for the addition of amino acids, and this is what's called the genetic code. And it looks something like this. Okay, so how it works is that there are what are called codons, which are little 3-segment nucleotides found on the RNA transcript. Those 3 different segment nucleotides, they code for different things. Okay, so for example, this one right here, GGC, if I was wondering what that was going to code for, I could take a look at the genetic code. The first letter in the codon is called G, so that would be here. So I know it's going to be something in this column. The second letter is G, so that would be, here's the second letter. G is over here, so I know it's going to be something in this column. And then the third letter is C, so that would be this one, GGC. If there was an RNA transcript bearing GGC, such as this one we see here, and it passed through a ribosome, it would lead to the addition of this amino acid, which is glycine. Okay, so these little letters here, they represent the amino acid that is going to be attached based upon this code that is read. The different amino acids that are out there are right here. Okay, and so each one of these little letters that you see corresponds to one of the amino acids. And remember that protein is really just a big long chain of amino acids. And so the chain of amino acids is determined by the sequence of these little codons that are found on the RNA. All right, so we said that glycine is the one that was added if it reads a GGC. Glycine is right here. And there's a whole bunch of different amino acids. There are 3, 6, 9, 12, 15, 17, 18, 19, 20 different amino acids. And they all get assembled in different combinations to make nearly an endless amount of different kinds of proteins. All right, so that would be our GGC. And then the next one that we see here is GUU. So that would be GUU. That's this 3-letter combination here. So that would code for the addition of valine, a different type of amino acids. Okay, so this is kind of the link. One thing that's interesting is that when we get to translation begins, meaning the addition of amino acids. It doesn't begin until one of these codons is read. The AUG is considered the start codon. And so I want you to appreciate something a little bit more complex here. On our DNA template, we have the TATA box. That's right here. This is what would first recruit the DNA polymerase, and then it would continue transcribing. But not all of these letters in the transcript might get used to make protein. The place where translation occurs, where the ribosome is actually going to bind and start reading the mRNA transcript, begins right here at this AUG codon. AUG is going to signal the production of methionine, and for whatever reason, this amino acid is usually the first one. It always begins with methionine. Okay, so we would know that this is what we could call maybe the start codon. And then then translation will proceed for anything downstream of the start codon. So there would be no amino acids added for this part of the mRNA transcript, because only anything downstream of the AUG codon is going to be translated. And this will continue and continue and continue until one of the stop codons is reached, and then that will signal the termination. Okay. Translation is all about proteins, and so if you need a little bit of review about how they work or what they look like, you can find Chapter 3. In brief summary though, remember that a protein is just a long combination of amino acids. It's got a primary structure, which is literally just the chain of amino acids, so each one of these things is an amino acid. If it's a long chain of amino acids, we call it the primary structure. Based upon the chemistry of the different amino acids, the long chain of amino acids can fold in on itself to form a secondary structure. The secondary structure usually is characterized by these two different shapes that form. There's the alpha helix, which is kind of a spiral shape, and then there's what's called the beta pleated sheet. And then it can form a tertiary structure, which would look something like this, where you've got maybe some beta pleated sheets and some alpha helices, but then the whole thing is starting to fold into a three-dimensional shape. And then once it reaches this three-dimensional shape, then it can actually have its functions. The three-dimensional tertiary structure is made because of the fact that there are side chains on each amino acid. Keep in mind there would be a long chain of amino acids. Well, as I showed you, the 20 different amino acids have different functional groups on them. Like one amino acid might have these groups on them, another amino acid might have these groups on them, and together those two are attracted to one another. And this one, all of these amino acids would also have functional groups. They're just not shown for simplicity's sake. And there's lots of different ways that neighboring amino acids can link together. These are called side chain interactions. They help form the tertiary structure. And then a quaternary structure is when two or more tertiary proteins are associated with one another. All right, so that's a little bit about protein. Now the actual process by how it's made is called translation. Translation's got 3 steps to it, just like transcription. There's initiation, which always begins with the AUG codon. Okay, so this is something that happens again at the ribosome, and so this is a picture that's kind of showing translation in action here. The ribosome, how it works is there's 2 parts to a ribosome. There's what's called the large ribosomal unit, and there's called the small ribosomal unit. And then here in this picture we see our little figure illustrating a ribbon of mRNA or mRNA transcript. Well, remember that the mRNA transcript was made in the nucleus, if it's a eukaryotic cell, and it traveled out into the cytoplasm and it found a ribosome. Well, here's the first, there could be lots of different codons here, but unless there's an AUG, this whole process doesn't begin. So AUG is the start codon. It's the one that's going to initiate transcription. How it works is that the mRNA transcript will pass through the ribosome in the 5' to 3' direction. These again just kind of give us some directionality to the molecules. As it passes through, tRNA molecules that have what's called an anticodon on them will bind to the codons. So here is why tRNA is unique. tRNA's got a part to it that can bind to mRNA. It also has a part to it that can carry an amino acid. When a tRNA molecule is carrying an amino acid, it's called charged tRNA, and it can release its amino acid, and then it's no longer what we call a charged tRNA. And there are 20 different tRNA molecules corresponding to the fact that each tRNA molecule binds a different amino acid. Actually, I maybe have misspoke here, but if we go back to the genetic code, there's probably not 20. There are there are 20. There are a whole bunch of different tRNA molecules, and each one is going to have a different anticodon. So the reason why this specific sequence here on the genetic code codes for this specific amino acid is the fact that out in the cell somewhere there's some molecule of transfer RNA that has an anticodon that binds to this specific codon, UUUU, and when it binds to that codon, it bears with it a phenylalanine, or this particular amino acid. So, like over in our picture here, the reason why this codon, AUGG, codes for the addition of the tryptophan, which is TRP, I think, is because there's a specific tRNA out there that has the anticodon that binds to UGG, and when it does, it carries with it TRP, the amino acid. Okay, so what will happen is as this is passing through, these codons are going to be exposed to these 3 different sites. There are 3 different sites here on a ribosome. The incoming tRNAs always come in through the A site. Then the amino acid that's between the P site and the A site will link together. A peptide bond will be formed, and then the whole mess of it will shift over one. And when it does, then I think I usually remember E stands for exit. The transfer RNA that's now no longer charged because it's lost its amino acid is going to be ejected out, and then they can go pick up another amino acid. So that's kind of all the process of elongation, forming peptide bonds. That whole process is a little hard to understand unless we're seeing it in action, so I've got a movie here I'll play in a little bit to help you visualize that. But the big idea is the fact that there are different transfer RNA molecules that have different anticodons. The anticodons bind specific codons, and the transfer RNA molecules with the specific anticodons have specific amino acids that they carry with them, and that's what enables this code to be meaningful. Okay, the final thing that happens then is termination. This is when a stop codon is reached, so this mRNA will continue passing through here until there's a stop codon reached. There's 3 different stop codons I showed you earlier on the genetic code. Once one of those reaches the ribosome, then the small subunit will break off from the large subunit, and translation will be done, and there will have probably been a really long growing chain of what we'd call polypeptide that was produced through this whole process, and then that would break off, float away free, and then there would be a protein that was made. Okay, once that protein is made, it can go through some protein folding and modification and targeting. So protein folding usually occurs naturally. It's determined by the sequence or the chemistry of the amino acids. So as soon as a big long chain of amino acids breaks free from the ribosome, it will fold up naturally based upon the chemistry of those side chains. There are things that are called chaperones, which can sometimes help to aid protein folding or transport. Proteins are given kind of what we could call a train ticket, which is called a signal sequence. The signal sequence is basically a part on the protein that is given to it to help the cell know where it should go in the cell. So for example, we have an RNA transcript made here. It travels out to the ribosomes and it makes protein through the process of translation. Well, this would be what an unfolded protein would look like. It would just be a long chain of amino acids, kind of like this. And then it folds up, it looks like this. And then usually on a protein we say that there's an N-terminus and there's a C-terminus and an N-terminus. This results by the fact that on like a peptide bond, when amino acids are being grouped together, all amino acids have this common shape to them, and here would be the side chains right here, and when they get linked together, it's formed a peptide bond which looks like this. The hydroxyl group of one amino acid and the hydrogen of another amino acid are removed through dehydration synthesis to create a peptide bond. This can be a very, very, very long chain that just keeps going and going and going, but regardless of how long it is, the amino acid that's over on this side is always going to have an N exposed, and the amino acid that's over on this side is always going to have a C exposed. So this, the one with the side of the C exposed, would be called the C-terminus, and this, the side with the N exposed, would be called the N-terminus. Well, usually on the N-terminus there's a signal peptide, just a series of amino acids, one after the next, which will help the cell read this protein and figure out where it should be brought to in the cell. All right, let's try a review question, and it'll hopefully make a little bit more sense as we go. What would be the sequence of bases on an RNA transcript formed from the following DNA template? Okay, so imagine somewhere in the genome there is this sequence of bases on one of the strands of DNA, and we're wondering what the RNA transcript would be made from that template. Go ahead and pause the video and see if you can work through this, and then I'll go through the answer here in just a minute. Okay, the answer is not too bad. You just have to, it just requires you know what nucleotides are added complementary to the template nucleotides. Okay, so here's our Tata box. This is where the different transcription factors are going to bind, and then once they're all bound, they're going to recruit the the RNA polymerase. And once the RNA polymerase is recruited, it will kind of go through and add all of the different nucleotides. The nucleotides that it will add are complementary. C's always bind with G's, so they would add a G here. It would add a G there. It would add a C, a G, An A, or yep, and then a U. Remember that this would be a T if it was DNA, but because it's RNA, it's a U. U's always replace T's. A G, T. Almost made a mistake there. U, C, 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 G, A, U, C, U, C, C, A, U, G, A, U, C. Okay, I did that kind of fast, but hopefully there's no mistakes. So that would be the RNA transcript. And you could probably include these here too if you wanted. Okay. Then the next question is a little bit tricky. Imagine that this, the RNA transcript, this whole thing would be called an RNA transcript. Imagine that whole thing when it's made its way to a ribosome and was translated. And assume that there wasn't any big modifications due to mRNA processing. Then the question would be, list the amino acids that would result, the polypeptide that would be made from this RNA transcript as it goes through the ribosome through the process of translation. This is a little bit of a trickier question. Go ahead and pause the video. I've included the genetic code here to help you answer this. Try and work through it, and then I'll tell you what the answer is. All right, the way to think through this one is to first find this start codon. AUG is the place that translation always begins. So if we're scanning through here, there is an AUG here, but the AUG should happen downstream of the TATA box. Okay, this is the TATA box. Usually how it works is the TATA box, then there's a couple of nucleotides, and then the start codon. So there's another one that happens right here, AUG. So this would be the start codon, and then that means all of this stuff right here is not translated into a polypeptide. We'd probably call that an untranslated region. Okay, and then everything downstream of this we could chop up into codons, which are again groups of 3. So the first one would be methionine. That methionine would get added to whatever was brought by the transfer RNA molecule with the antisense codon to UCC. So if we look at the genetic code, we see, let's find UCC. Right here. UCC would be the thing that codes for the addition of serine. So serine would get added. And then we have CGA, that codes for CGA. That codes for the addition of an amino acid called arginine, ARG. And then we have UCU. So we go to UCU. That also codes for serine. And then we have CCA. So let's find CCA. That's going to code for the addition of proline. And then we have UGA. So if we go to UGA, oh, that is a stop codon. All right, so we would say this is a stop. And there is no amino acid added here. So usually when we look at our translatable codons, the number of amino acids is going to be one less than the number of codons there are. The number of codons we have here is 1, 2, 3, 4, 5, 6. The number of amino acids we have is 1, 2, 3, 4, 5. This doesn't bring an amino acid. So at the end of the day, it's going to say list the amino acids in a polypeptide that we made from that RNA transcript. Well, this mRNA transcript, after it's been translated, would produce a link of 5 different amino acids: methionine, serine, arginine, serine, and proline. That would be released from the ribosome. It would maybe fold up into a structure and then act as a protein after that. This would be a very, very small polypeptide. Most proteins are several hundred amino acids long, but just for the sake of illustrating how this works, we made it small. Right, so do you feel comfortable doing something like this? If not, watch through this a few times and then practice through it. You should be able to do this question without looking at the answers, and then grade yourself afterwards and see if you get it right, because there will be a question on the exam assessing your ability to do this. All right, here's a little video that shows this process. And again, this is probably the most helpful. If anything I said on this video, this is probably the most helpful, because it really helps to just see this in action.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
Okay, so that's a really great video to help you kind of see in 3D what's going on. Here's another review question for you. Go ahead and pause the video and see if you can think your way through this one. All right, the answers, let's go through them here. All right, the answers, let's go through them. The first one says that an RNA polymerase binds to a promoter upstream of a gene. Well, we know we're talking about RNA polymerase, so that's got to be transcription. Transcription is the process that makes mRNA from, or not necessarily M, but makes RNA from DNA. So if we're talking about RNA polymerase, the thing that literally builds the polymer of RNA, that's got to be transcription. And then we just have to figure out, is that initiation, elongation, termination, or mRNA processing? because it's binding to a promoter that assumes initiation, so that would be B, correct answer here. Peptide bonds are formed between amino acids. We're talking about amino acids and peptide bonds, so that's translation, because translation is the thing that makes protein. And specifically when the amino acids are linking together, that is elongation. The peptide, the polypeptide, is getting longer and longer and longer, so that's what I mean by elongation. G is the correct answer. DNA polymerase adds nucleotides to a growing strand of nucleic acid. Here it's the DNA polymer ACE, so it's the enzyme making the DNA, so that's got to be DNA replication, which is what we talked about in an earlier chapter. Okay, a stop codon enters the A site of a ribosome. If we're talking about ribosomes, we're talking about protein. A stop codon would make that terminate, so that would be the termination of translation, H. Bases of guanine, cytosine, adenine, and uracil are added to a strand of nucleic acid. It doesn't specify whether this is RNA or DNA, it just says nucleic acid, which could be RNA or DNA, but the fact that it says uracil lets us know that this is in fact RNA, so that would be elongation of transcription. If, however, it said not uracil but thymine, then that would be replication of DNA replication. Primase adds an RNA primer, that is a component of DNA replication. The small and large ribosomal subunits attach around an AUG codon. That is going to be the first step in translation. Remember, if you see ribosome, we're talking about protein, so it's probably translation, so that would be initiation. Introns are removed, that's part of mRNA processing. Ligase creates a continuous strand of nucleic acid. Ligase is something that's involved in DNA replication. Alpha helices and beta pleated sheets are formed within a polypeptide chain. That would be a post-translational folding, meaning after the thing is translated, after the protein, the polypeptide is released, how does it fold? All right? Okay, I put this slide up because how genes are expressed. The word gene expression means the actual transcription of a gene and translation of the gene. So, you know, you've got lots of genes in your genome, but some of them aren't being used right now. But in another moment, they might be used, and when they're used, that means that the RNA polymerase is going to bind to the promoter, transcribe the gene, and then the transcription will be processed and then headed over to the ribosome. So how to turn genes on and off, that's the word gene expression. So if there's a gene with a lot of gene expression, that means that the gene is being transcribed and translated frequently. If there's a gene with low expression, we would say that's a gene that's not making as much RNA and consequently not as much protein from the gene. Well, the cell can regulate how to turn on and off gene expression, and these are kind of the four principal ways. This is all of chapter 16 summarized in one slide, so I won't go into too much detail, but basically a cell can control whether or not the gene is even transcribed in the first place. This would involve the transcription factors. Some transcription factors are silencers, so they actually keep the gene from being transcribed. Some transcription factors are activators, so they'll recruit the polymerase and have that gene transcribed. And even if an RNA transcript is made from a DNA template, there can be some things that happen post-transcriptionally to prevent that transcript from making its way to a ribosome and being translated. Also, translation can be modified. Either the ribosome can be shut off or turned on, or different things can happen during the whole translational process to either release the polypeptide or stop the polypeptide from being made. And then even after, if the RNA transcripts made it to the ribosome and the ribosome has translated that transcript and made a protein from it, sometimes the protein itself can actually be modified, either inhibit or broken down or turned on, or something can be added to it to make it not functional or to increase its functionality. That would be post-translational regulation. So lots of different steps along the way where the cell can intervene to either reduce or boost the expression of the gene. The last thing we'll talk about in this lecture is mutations. What happens when, what is a mutation and how does that affect this whole process? There are two different kinds of mutations. One's called point mutation and another is called frame shift mutation. A point mutation is just what happens when one nucleotide is added incorrectly, and that the frame shift is still intact. Meaning, remember when we said that there are, there's all of these nucleotides in a strand, in an RNA transcript, and they are broken into codons. Well, the codons are going to be the frame shift. So the frame shift right now, or the frame right now, is showing that these three letters are one codon, these three letters are another codon, these three letters are another codon. But what if we were to remove or add a nucleotide? That would change the frame shift. So now it's maybe like this. Okay, so that's what we mean by frame shift, is when what do we regard as the codons for each amino acid. Okay, well, in either case, frame shift mutations will cause a shift in the frame, reading frame. Point mutations will not. There are three different kinds of ways that a point mutation could lead to, or three different kinds of point mutations that it could occur. One is called silent, because there is actually no effect. And usually this happens in the third letter of a codon, because the third letter of a codon isn't as important as the first two letters are. So in this example, we're seeing here that what should have been transcribed was this, but for whatever reason there was a mutation that occurred on the DNA template. So instead of an A being put here on the RNA transcript, a T was actually placed here. This would happen, again, if the DNA template for this would be a C, this is actually showing you the DNA template. I know that because there are T's here, not U's. Okay, so if the DNA template was GTA, that would code for our transcript, which would lead to the production of the addition of valine. Well, if that A was accidentally converted to something and a T was put there instead, even though it's a different codon, this is GTT or this is GTA, both of these codons code for valine. So there, in the end, is no really consequence of that DNA mutation, because the same polypeptide will result from it, which will lead to no really gain or loss of function. Usually, whether a mutation is harmful or not just depends on if it changes what the protein is built from the mutation. So that's silent. A missense means that leads to one amino acid substitution. So in this example, the DNA template should have been CCC, but for whatever reason there is a mutation that happened and it led to an A being put in here. So instead of a proline being added, a threonine was added. So there's actually been a substitution here. Whether that substitution is significant or not kind of depends on the importance of the amino acid. If the amino acid that has been substituted results in a really, really different chemistry, leading to a different folding to the shape of the polypeptide and a different function of the protein, then that mutation could be pretty detrimental. If it leads to the insertion of another amino acid that kind of has a similar chemistry than the one that was originally intended, it could be that once that polypeptide is made, it still folds the same way and it has kind of the same function, so it's not as detrimental. It all just depends. Okay, then a nonsense mutation is one that leads to a premature stop codon. So in this example here, in the DNA template there was a TAC, the C got converted to a G, and then that led to a premature stop codon. This can be a very detrimental mutation, because one little change in one little base pair can lead to could be a catastrophic change in the protein that's supposed to be made. And if it's an important protein that needs to be made in the cell and the cell no longer can make that protein, then that could lead to some serious complications. Another type then is the frame shift mutation. What happens here is usually it's an insertion or a deletion of a base, not just the swapping of a base, as was the case here in the point mutations. And in a frame shift mutation, if let's say in this example we had a GTA, and then the T and the A were deleted. So now we have a G. These mutations just slid up. So now we have GCC. Well, that suddenly changes the whole reading frame. And even though there's only two bases that were affected, because the whole reading frame has been changed, everything downstream of that mutation is going to lead to a different array of polypeptides being added because the reading frame is now changed. So that can have a very detrimental effect as well. That will conclude our lecture on DNA and protein.